So the outline today, we're going to have two really great talks. The first on adaptive trial design with Phil Spinella, the second on innovative analytic methods in clinical research with Jen Mazinski, and then we'll have a question and answer session really focused on whether or not RCTs are needed, and that's going to be moderated by Vince Faustino. So for our first talk, I'm going to stop sharing Phil so that you can share. So Phil Spinella is a professor of pediatric and surgery in the Division of Critical Care Medicine and co-director of the Trauma and Transfusion Medicine Research Center at the University of Pittsburgh. Does a ton of research and really an expert on this topic.
Thanks, Michelle, for that intro and the opportunity to speak with the BloodNet group today. Let me see if I can get this someplace else. There we go. So I'm going to spend about 20 minutes talking about adaptive trial designs and with that, Bayesian analyses too. This talk, and as what I've given it in the past, has been, relative to life-threatening traumatic hemorrhage, but these concepts and principles can be applied to any subject matter, especially within critical care medicine. And I think moving forward, right, we have to, especially in pediatric critical care, have to think about designing our trials where we are working smarter, right, and not harder. We all work hard already to get trials done. We don't need to make it harder on ourselves. And it seems like, we are still, in general, doing things the hard way. And I'll get into that a little bit. And it's time to start changing the paradigm. My disclosure slide, nothing at all relevant to this topic. Oops, backwards. So I'm going to compare and contrast the classic clinical trial design and frequent statistics to adaptive trial designs and Bayesian analyses. And then we'll talk through some, trauma trial, and we can definitely talk about critical care related trial applications and limitations of these concepts. So with classic trial design and frequentist statistics, right, the study design is almost always determined based upon non-randomized data. Usually it's observational data that we've collected many years before the trial starts, and it's many years because it takes a good 5 to 7 years between collecting the observational data, getting funding to do the pilot study for feasibility, completing that, then getting the funding, which could take a few years to do the definitive trial, and then starting the trial. And by the time you start the trial, you really have fairly old, stale data, and practice has likely changed over time too. So that is one of them, a significant limitation of kind of the standard approach. In addition, with a classic trial design, it's very rigid. You can't change during the trial from data that you're learning from the trial, the study interventions, the study population, and the doses of therapies. You're pretty much locked in to the study that you design when you start the trial throughout the entire period of recruiting patients. And that's a significant limitation. And usually, not always, but usually, we're only studying one intervention at a time. And as you all know, when we treat our patients that are critically ill, we often, no matter what their clinical condition is, it's a bundle of care that we're using for them, whether it's sepsis, ARDS, renal failure, severe bleeding. So if we treat patients with bundles of care, we should be studying bundles of care to see what bundle is best for what specific patients. And with the classic approach, we don't do that. We study one therapy at a time, and then we try to incorporate that into that overall bundle, which may give us different results. When it comes to frequentist statistics, right? We've all been, bowing to the 0.05 God for the forever. And when you think about that, and what does this actually, mean? A p-value of less than 0.05 means there's less than at least a 5% chance that the differences found could have occurred under the null hypothesis, or basically it's less than a 5% false positive rate. But that's, again, super rigid, right? And again, when we treat our patients in the ICU, we're normally not incredibly rigid about the decisions that we make. It's all very context responsive. So if you had a trial where the p-value was equal to 0.06, I've actually had studies where the p-value equaled 0.05. And if it equals zero point zero 0.05, according to frequency statistics, that is not statistically significant. It's going to be less than 0.05. But what does this mean? It means that there's probably a 6% chance of a false positive a false positive result. But in a certain context, that might be enough to change practice. So again, the rigidity of frequency statistics, I think, is a major limitation, and we should be more flexible in how we interpret data from trials. So again, inflexible is based on outdated data. You don't learn from the data. You don't learn from the randomized data that you collect during a trial, right? When you design a classic, when you take the standard approach for a classic trial design, that observational data is non-randomized data that you use to then inform your randomized trial design. Instead, with an adaptive design, which we're about to get to, it's actually randomized data that you're using to inform decisions made during the trial. And again, the stats approach is, I would say, too rigid and can lead to misinterpretation of data that's generated during a trial. So with adaptive trial design, this allows for prospectively planned modifications to one or more aspects of the design based upon accumulating data from subjects in the trial. This is designed to take advantage of the accumulating information. It, in effect, then reduces the uncertainty in the study design because you're using data that's randomized, that's being collected during the trial, which is not old data, which is not old experiences or in a different context in which you're treating the patient now. It allows for modification to key trial parameters in response, again, to accumulating data according to pre-specified rules. Okay, adaptive design doesn't mean you get to change a trial however you want to do it at that time. It has to be based upon a priori or pre-specified rules that were determined before the trial started. And key parameters can be modified throughout the trial, such as the subjects included or therapies, and we'll get into specific examples hopefully pretty soon. Again, by taking this approach with pre-specified rules, the trial can be very rigorous with well-defined operating characteristics. And I think probably most importantly, right, by using this approach, it increases the probability or chances that you're going to get an appropriate and valid, actionable answer at the end of a trial, right, which can potentially improve outcomes for our patients. I mean, those of us who have been around long enough have realized that pretty much almost every trial that we conduct in critical care and peds critical care is in the end not statistically significant, right? Most of our trials end up that way. And it's because, I think it's because of the things that I've been mentioning before. And we're not able to modify the study to then get at least an actionable answer versus enrolling everybody. There's usually a very heterogeneous population. And we don't try to figure out, well, in what patient population would this therapy work or not. Now, the FDA is in support of adaptive trial designs. If anything, now 6 years ago, 2019, they published their guidance for how to use adaptive trial design for FDA-regulated trials. Interestingly, the group that wrote these guidelines basically come from the Berry Consultants, which are I think, the premier group internationally that consult with many of us to design clinical trials. With adaptive, so what are some of the main methods? A platform trial is a trial in which multiple interventions and multiple groups of interventions can be studied at once. Again, this concept of studying a bundle of care to see what bundle works best for what patient population. Randomized adaptive, I'm sorry, response adaptive randomization allows you to change the ratio that you're randomizing interventions. We normally can start 1-to-1 with your randomization ratio, but if the trial results are indicating to you that one group is doing better than the other, if they have pre-specified rules on how to do that, you can now start to enroll more patients into the group that's doing better to accelerate getting to superiority and getting the study done faster. It also allows for more safety data in the new intervention too. Enrichment is a process at which, with pre-specified rules, you can change eligibility criteria. If you're noticing in a certain patient population, the therapy, the intervention is working better than an alternative population, you can now remove uh, patients from the trial that were not benefiting from the intervention. So a good example would be, let's say you're comparing whole blood to components. The data in the trial is telling you if the maximal amplitude, right, from TEG is less than 60, those are the patients that are doing better with whole blood. So with enrichment, you can now exclude patients if the MA is above 60, just for example. Rarely you can adapt your endpoint or outcome in an adaptive trial design. This often isn't done. But if you're not sure about what your primary outcome should be, and you can include them both, and then with this approach also change during the trial to name a specific primary outcome for the trial. Platform trials are probably the most commonly used adaptive method. Again, this allows you to compare more than one intervention against a control. The nomenclature with platform trials are, they use the terms domain and factors. A domain is a category of treatments. So for example, blood products, coagulation factors, antifibrinolytics are categories of therapies. Factors within the domain, for example, would be different types of blood products within that domain. And you can see the examples I have listed there. There's been a lot of interest in using platform trials for trauma trials. Our research group has collaborated with other international experts, and we've published these three papers to explain why they should be done, how they could be done, and the ethics related to platform trials in life-threatening bleeding. So if this is of interest to you, these papers are good references. Again, the advantages, again, that you can study a bundle versus one intervention at a time. It also leads to, I would call it operational and economic efficiency. When you design a platform trial for a certain condition, again, we can take life-threatening hemorrhage, you spend the money on the infrastructure, right, to set up that network, a master protocol that can be used to determine eligibility. And then once the first trial or the first domain, if you finished it, you can now bring in another intervention over time and not have to redo the entire study. The metaphor that people use is you don't build a stadium, right, for every football game in a season, right? We use the same stadium every Sunday, and you don't have to repeat that infrastructure. It's the same thing with platform trials. So here's an example on how a platform trial could work. Within one domain, you can have a standard of care control compared to three different interventions, over time on the x-axis. But then, what if new therapies come along as you're performing the trial? You're like, oh, you know what? There's some good data for therapy D and E. We want to study that too. This allows for the flexibility to bring in new interventions while the trial is accruing patients. If during your first interim analysis, right, as now these five interventions are being compared to one control, what if one of them is determined to be futile, right, or causing harm? That can now come out of the trial and allows you to now recruit all of those other patients into the other, uh, for those other factors to increase enrollment there. You're not wasting time with this futile or potentially harmful intervention. The trial continues, and now you find that one therapy, B, is actually superior to the control arm in the trial. If that occurs, now therapy B becomes the standard of care as the trial continues. So now you're comparing the other interventions to the intervention that was determined to be superior to the control group. So you can see how this allows for flexibility and to not only increase interventions that you can consider for the trial, as data is accruing during the trial, you can drop therapies or bring new therapies in. And if one is superior, the trial keeps going. Because what if with more patients in D, D could even eventually be better than B was, right? And allows you not only to evaluate individual therapies for superiority, you can still look at the bundle of care and the combinations of therapies as well too related to outcomes. I'm going to skip that just because I realize I'm getting a little bit late on time. When it comes to Bayesian statistics, right, instead of frequentist statistics, Bayesian analysis basically allow you to evaluate the statistical significance of the study in relation to the question being asked and the population being studied. It also is less limiting when it comes to what is significant or not. It's basically what you get is a posterior probability that one therapy improves outcomes more than the other. So you can get a posterior probability that, let's say, is 95% therapy A is better than B. It could also be 88%. The probability is 88% that therapy A improves outcomes more than therapy B. And depending upon the question being asked, you might say, that's good enough for me to change practice. So I'm going to give you two examples to try to highlight that concept. Consider a high-risk condition where mortality is 40%, life-threatening bleeding in children. And there's a very safe therapy that you're considering as your new intervention, has a very low adverse event risk. This new therapy is also inexpensive, cheap, and your standard therapy actually is not based on high quality data. If at the end of that trial, comparing those interventions, would you change practice if the intervention had a posterior probability of 70%? So 70% chance that therapy A is better than B when there's a high mortality, it's safe and cheap. We might do that. What about at 80%? Does it need to be 90% for you? And the whole point here is you get to make that decision. And there's not somebody else saying that a 5% risk of a false positive rate is the only way that you can determine statistical significance. Another example, what if it's a low mortality condition? High risks. It's expensive and the standard therapy is based on good data. Well, here, maybe now your threshold should be higher, right? Maybe you should only change practice if there's a 95% probability that therapy A is better than B. Whole point is this is context responsive and there are no absolute rules on what is significant or not. Trials have been done with the frequentist approach. This was the proper trial comparing blood product ratios in adult trauma patients. And when they went through the frequentist approach, which was their stated approach in the trial, p-value of 0.12 for 24-hour survival, 0.16 for 30 days. So the interpretation of this trial was that no difference, no statistical difference in ratios, and you should not change practice. Well, when they repeated this using Bayesian analyses, now the results are a high ratio had a 93% probability of improving 24-hour survival compared to lower ratios. So if that was the primary result in the initial paper, I think we all would have confidently said, well, that's significant. I would definitely change practice based upon that. So this is just a good example on how you can get very different interpretations based upon the analytic approach that you take. Um, Adaptive trials and Bayesian statistics can be used independently. They don't need to be married together, but they often are. Adaptive trial limitations are that the planning phase does require a lot of time to do a lot of simulations and modeling to come up with those pre-specified rules. Limited experts that can help you design these trials. It's kind of a small group of people that are experts at this, although that is growing. And funding mechanisms, while there are some funding agencies that are actually supportive and prefer these trials, I would say currently the majority are not because there needs to be some flexibility in the budgeting. Bayesian statistics limitations, you have resistance from the old school frequentists that kind of just don't like this non-rigid approach. Limited number of Bayesianists as well. And funding agencies, they in the past have been less familiar, but they're becoming more familiar with it over time. And I've shown you the FDA is supportive of this approach too. So in conclusion, these are adult trauma trials that all studied one therapy at a time, right? Using frequentist approaches. We need to work more intelligently and stop working so hard getting single intervention trials done. We should be really thinking in critical care and specifically peds critical care platform trials where we can analyze a combination of therapies in our critically ill patients. This approach is scientifically efficient, time efficient, cost efficient, etc. And I'm done. Thank you. Sorry for going a little fast, but I'm already 4 minutes over.
Oh, we're doing okay on time. Thanks so much, Phil. If you guys have any questions for Phil, please put them in the chat, and then we also have like the question and answer session at the very end too. So with that, I'd like to introduce Jen Musinski. And Jen, you can start sharing your slides. So Jen Musinski is an associate professor of pediatrics in the Division of Critical Care, and also associate chief clinical research officer for research informatics at Ohio State University and nationwide. So in addition to being a really awesome person, also an expert on these topics. So excited to learn from you, Jen.
Absolutely. Thank you so much, Michelle. Thank you for the opportunity to speak this morning. My one caveat here is I'm mushing two rather big topics into 20 minutes. So this will be, by definition, a high-level overview. And depending on your preferences, this could be good or bad. There will be no math discussed here. Wouldn't know a lot of the math myself anyway. But basically, I want to talk a little bit about machine learning and introduce the concept of real-world evidence as a way to complement some of the challenges of randomized controlled trial design that Phil just mentioned. I have no financial conflicts of interest. I do have some NIH-funded funding that is related to what we will be discussing. So anyone who has stepped foot inside an ICU probably appreciate the fact that we live in a data-rich environment. We make clinical decisions in a data-rich environment. Every day, we are, as clinicians, integrating various streams of data in real time, making decisions, sometimes on a minute-to-minute basis at the bedside. And so I think that begs the question for many of us, well, what if we could really leverage that treasure trove of data that are being generated every day when we take care of our patients in order to learn more about how to take care of them better? What if part of our research armamentarium is the data itself that we are generating every day? And so what we're talking about is big data. I think we can safely say that we are now in the era of big data. It's not coming, it's here. There, of course, have been lots of definitions of big data through the years. And so I do think it's important to think about what we mean when we say big data. This is a relatively recent review article that I think outlined this nicely in pediatric research from a couple of years ago. And talks about the features of big data and what we mean when we're trying to capitalize on these larger data sets. One, of course, is volume. When we say big data, we mean big. But the other is often a variety of data. And when we talk about multimodal data sets, so these may be data sets where you're combining structured data elements from the EHR, things that are those discrete elements of data like demographics or vital signs or medications or laboratory data, along with perhaps unstructured data, so natural language processing applied to progress notes as an example, imaging data, high-dimensional waveform data imported directly from our bedside monitors. Oftentimes now when we're talking about large data sets, we're talking about merging multimodal data from multiple sources, not unlike what we do at the bedside. This brings with it that question of velocity as well, or that high dimensionality, not just in the number of elements, but the frequency of data over time. Certainly, if you think about the frequency of data that it takes to regenerate a waveform on a bedside monitor, that's very different from a vital sign number that is collected once an hour. And that obviously, exponentially increases the amount of data that we're talking about and speaks to the types of methodologies that need to be applied in order to process and analyze those data. Variability of data over time, the extent to which data are consistent over time, continue to be reliable over time, which brings us to the veracity of the data as well, which I think is really important. We'll touch on this in a few slides, where we're talking about the difference between data that are collected under the auspices of a research protocol where people are trained to collect data prospectively versus the data that happen in the real world, and the extent to which we can have high quality data that are accurate, reliable, and reflect what we think that they do. And this is where having very close collaboration between clinicians and data scientists to make sure that we are interpreting those data elements the same way becomes very important. And ultimately, this all rolls up to value. What value can be extracted from the data? What meaningful conclusions can be drawn from the data that we are collecting and analyzing? Specific to critical care, this is a really nice review article that I share with folks pretty frequently that was written specific to pediatric critical care medicine and thinking about not only the different data sources of data science in critical care, but what are the use cases for data science in critical care? And the nice thing I like about this paper is that it helps break down those use cases into three main categories. And here we're thinking specifically of machine learning and what one might have called AI before generative AI. We're going to leave generative AI to the side for a moment. But the use case number one, or category use case number one, is I have a group of patients, some of which have an outcome, others don't, and I'd like to predict who has that outcome. So can I predict cardiac arrest? Can we predict patients who are at risk for acute deterioration? Can we identify sepsis earlier with a machine learning algorithm than we might as clinicians? Can we predict an outcome that allows for prognostic enrichment for a study so that we know that we have the right patient population in our randomized controlled trial when we do one? There are other categories of machine learning where I'm not trying to predict an outcome that I know, but I'm trying to understand the extent to which there are heterogeneous populations within my diagnostic cohort. Phil touched on this a little bit already, but we know that many of the diagnoses that we apply to our patients in the ICU are somewhat syndromic diagnoses, and we probably are mixing together patients that have distinct pathophysiology. This has been shown several times over in ARDS, sepsis, and is probably meaningful when we think about, does it really make sense that one therapy applied to that entire population would work the same in everybody who has very different underlying pathophysiology? And can machine learning help us to identify who those patients are and what those subphenotypes might be? And then, of course, there's image processing. The extent to which perhaps could machine learning or AI identify features in radiologic images that augment the human interpretation and can provide even more information than what we otherwise would have. So specific to transfusion, over time, there have been increasing publications applying machine learning or AI to transfusion medicine. For the most part, many of these actually are predictive algorithms to predict when patients receive transfusion, whether it be in the setting of trauma or operative medicine. And this is a really important question for resource planning, for blood product allocation. But I have a slightly different question. I'm really interested in, can we predict when a patient should be transfused rather than whether a patient is transfused? And it's a question I've had for a long time, and a question that at times in my career, folks have discouraged me from pursuing because it's too complicated. And in fact, it is complicated, and I'm stubborn. Maybe not quite as stubborn as Dr. Spinella, but stubborn nonetheless. And I continue to pursue this question. So the point of this slide here is, and this comes from a review article written a few years ago, that tries to put together in one map, or brain map, if you will, of the things that either we do or perhaps should think about when deciding whether to transfuse someone. And the point of this isn't to follow all of the arrows, but to understand that, yes, it's complicated, that what we're trying to understand when we make a decision of should I transfuse this patient or not, it's complicated. And yet our evidence base up till now has largely been based on transfusion or hemoglobin-based thresholds for transfusion. I imagine that most on the call are familiar with the TRIPICU study, and this is a fantastic randomized controlled trial that really set a new standard, really did change practice for how we transfuse children in the ICU. When my residents or fellows say, "Oh, hemoglobin threshold of 7," and sometimes I ask them, "Okay, where'd that come from?" Every once in a while, somebody will point to this. Oftentimes, at this point, they're not even sure where it came from. But that really set a new standard. One of the limitations, however, of not just this trial, but of any randomized controlled trial, when you think about what a randomized controlled trial is and how we analyze the data and what assumptions go into that trial design, there's a very fundamental assumption that when you apply a therapy to your sample population and you measure the effect of that therapy, that effect is largely the same across your population, because what you get at the end of the day is an average effect. If I'm treating an individual patient, I don't care so much as a clinician about what the average effect is. I know what is the effect for my individual patient. And if I know that the reality of the ICU is that there are different patients in my population, and it's highly likely that within that population, there are some patients who might benefit, some patients who may actually experience harm, other patients for whom there is no net effect or no effect, the net effect of a trial could be that there's no difference, but that maybe isn't as meaningful for individual patients as we would like it to be. And so this brings up a challenge of how do we identify those individual patients for whom red cell transfusion would be beneficial versus potentially harmful. This is something that we're trying to tackle now with the transfusion organ dysfunction and pediatric septic shock and PROPIX trial. I won't go into too much detail, but just as a brief overview, this is an ongoing 17-center observational study where we are looking to use machine learning techniques to try to do exactly that, to identify patients for whom transfusion is likely to be beneficial versus those for whom transfusion may be harmful. We have two aims. One is to use clinical variables, focusing largely on hemodynamic variables, which we hypothesize to be important in this patient population for this question, and then aim to use a host of protein and RNA biomarkers to define immune and inflammatory phenotypes, and then determine the extent to which those phenotypes might change that prediction of who might respond favorably to transfusion. This study is ongoing. We are up to 484 plus patients. That number's a couple of days old. So we've passed our 2/3 mark and are hoping to wrap up enrollment within the next year or so. So I hope to have some more information to inform this question relatively soon. Now I'm going to take a left turn altogether right now. Somewhat. So we're still talking about observational study designs. We're going to talk about a little bit of a different use case for observational study designs, and this is real world evidence. And I thought that this would be a nice complement to the talk about randomized controlled trials, in two parts. One, because real world evidence can complement some of the challenges of RCTs, but also we are seeing more and more studies utilizing real world data and real world evidence, which is likely reflective of the fact that the FDA now has mechanisms to incorporate real world evidence into regulatory decisions, and that space is continuing to evolve. So what do I mean by real world evidence? Well, I'm going to refer again to the FDA. This is their definition. So real world evidence is clinical evidence that is derived from analysis of real world data. So of course, now we have to know what is real world data. Basically, is real world data are any data that exists that pertains to a patient's health status and or the delivery of healthcare. And the forms of real world data that are mentioned or examples are anything from electronic health records to claims data, registry data, digital health technologies, the wearables that we all have that are collecting data all the time that may also pertain to our health status. So why would we think about using real world evidence? Well, Phil already outlined some of the challenges of randomized controlled trials. He talked about the fact that we have multiple simultaneous treatments and multiple interactions that are really hard to recapitulate, even in a platform trial, and can real world data and real world evidence help complement that, help address that limitation. We know that patient trajectory is important. That's also a little bit hard to incorporate into a trial design. Our patient numbers within pediatric critical care, in some respects, depending on what diagnoses we are targeting, we are in the rare medicine space, or rare population space. And so our studies require multi-center collaboration that's highly resource intensive from a time and cost perspective. We have ongoing challenges in our field with low enrollment, concerns very real about potential differential enrollment across populations and the extent to which our randomized controlled trials are reflective of our overall patient population. And then Phil also had touched on the patient heterogeneity as well. And making sure that we can fully incorporate that into our study designs. And so again, it begs the question, well, we're already generating this much data. Can we use it? And can we use that to learn how to treat our patients better? And so the answer to this question is yes, with an asterisk. There are, I think, some very real and fabulous opportunities for the use of real world evidence as an augment to randomized controlled trials, but there are some challenges as well. And some of those challenges, I think, are actually amplified within the ICU setting, and this is one of them. So when most folks will talk about observational comparative effectiveness study designs or generation of real world evidence, they're often talking about taking observational data and creating essentially either digital twins, or they talk about the digital counterfactual, where you're trying to emulate a randomized controlled trial as best you can within an observational study design, whether that's using a propensity weighting or other methodologies. The really important caveat is that those statistical methods are only as good as the data that you've collected and the extent to which those data actually reflect what we use as clinicians to make the decision to apply a particular therapy. So in other words, because a lot of times when we're trying to study something like transfusion or many of the treatments in the ICU that are what I might consider a step up therapy, right? So by definition, the sicker patient gets the therapy. It's sometimes really hard to recapitulate that in an observational study design to know why did I transfuse this patient and not this patient and get around that confounding by indication bias. And so to drill this home a little bit, I want you to consider an 8-year-old, previously healthy child with group B strep, sepsis, and a hemoglobin of 8. And another, their digital twin, same diagnosis, same demographics, same hemoglobin. This patient gets transfused, the other patient doesn't. We do our study. We find that transfusion is associated with adverse outcomes. Okay. Well, maybe we didn't capture all of the variables. We don't know anything about the hemodynamics. Okay. Well, walk in the door, the hemodynamics were the same. Okay. Well, what about their trajectory? And this is one that it has been, I think, historically challenging to capture, but really important, right? Because we know at the bedside, this patient here who gets 60 mLs per kilo of fluid and stabilizes versus the patient down here who gets that much fluid, is on an epinephrine infusion that's escalating, has gotten stress dose hydrocortisone, is clearly a different patient. And yet, depending on how we design our study, which variables we use in our study, the extent to which we're incorporating longitudinal data and trajectory, we may not capture that in our observational study design. And so the point of this slide is that I think there are very real opportunities, but some very real challenges that could lead to potentially inaccurate interpretations of our data if we're not really careful about our study design. And I also think, I've said this a couple of times over, but it bears repeating, that because of some of these challenges, that it's highly likely that at least now, the best use of real world evidence really is as a complement to, but not entirely replacement of the RCT. You just can't fully recapitulate the RCT. But you can do things to make RCTs run a little bit better. Or to augment the planning of those trials. Phil mentioned the 5 to 7 years of observational need to plan a trial. If you have a real world data infrastructure built, maybe you can shorten that to a few months. And that's one practical way that these two modalities can really work together. Now, that said, one of the challenges of real world data is that it is sometimes messy. So compared to, again, you have your prospective study where you're actively managing how the data are collected and monitoring the quality along the way. When it comes to real world data, you're left with what's in the real world. So sometimes it's fragmented. We still are working on trying to standardize electronically curated data across multiple institutions. There certainly are efforts afoot, and a lot of progress has been made. Things like the NIH Bridge to AI program, the PICU Data Collaborative, PedsNet, and others that really have done a lot of work in this space, but there is still work to be done in trying to pull together data and harmonize them across multiple institutions. That being said, the opportunity here is when we are able to do that, we can access a lot more data than you possibly could get by having clinical research coordinators look at the EHR and use that to fill in a case report form. Generalizability can sometimes be a challenge. Here, the variation in care across settings can be a plus and a minus. Sometimes that variation in care is what you're able to leverage in order to identify potential comparative effectiveness, though that variation in care in all other aspects of care can also confound those comparisons. The flip side of that, of course, is that potentially real world data can help to mitigate some of the potential concerns of underrepresentation in randomized controlled trials and make sure that our interpretations are generalizable across the populations that we treat. And of course, there are strategies to help improve that. I mentioned briefly that the FDA now does have some frameworks that enable the incorporation of real world evidence into approval decisions for drugs and devices. I'm not going to spend a lot of time there. If there is interest, I would refer to FDA guidance on that. But this is still an evolving category and an evolving space. And so anyone who is in this space is, constantly having to re-understand the rules of the road, if you will. And then the time and the cost, not unlike what Phil described for platform trials, one of the real barriers here is that the time and cost is largely in that upfront investment. It's building the data infrastructure, building the data governance upfront, which is different from how we typically think about funding science, where it's funding individual projects. Here, you're funding the infrastructure. The individual projects then are much more feasible and efficient, but it does take a lot of upfront investment, and somebody has to put in that upfront investment. And of course, there are some practical strategies. And as the methodologies for doing this become more efficient, that upfront investment can hopefully be lessened over time, but it still is significant and can be a barrier to entry. And then when we think about the outcomes themselves, important to note, of course, we're certainly more and more interested in those post-hospital discharge outcomes. And this is really challenging to capture in real world data, particularly when we're in systems where it may be hard to follow our patients longitudinally, where you have loss to follow up in the electronic health record and you don't know why, where you may or may not have those outcomes actually detected, things like hospital readmission as an outcome, depending on what your data source is. Do you miss someone who's gone somewhere else who has, if you're using claims data, has switched their cures and things like that. So there are definitely some caveats, concerns, but there's also a potential. And one of the uses from the FDA's perspective of real world evidence, of course, that we've used all for a long time is continuing to track potential adverse events after RCTs are completed. So those in-market studies that we've been familiar with for years. And again, just the caveat that I don't want to suggest that real world evidence would replace the RCT, but I really do think that there are ways that we can work together and that the pluses and minuses of each really do complement each other. And so sort of looking ahead to the future, I do think the extent to which we really can capitalize on building infrastructure to have robust, reliable, electronically curated data that are able to be harmonized across institutions, that are multimodal, that preserve temporality, which is one of the huge caveats and limitations in our work for using some of the registry data and the claims-based data, is the lack of granularity about temporality, which is, of course, important for causal inference. And really, at the end of the day, it's about, I think, coordinated interdisciplinary efforts. The way that we can have observational study designs complementing RCTs is by having clinicians, clinical researchers in the observational space, data scientists, informaticians, randomized controlled trial or trialists, all working together towards similar questions. And I think we can all get there better and more efficiently together. And that's all I have.
Thanks so much, Dr. Musinski, for the clarity and making it very motivating and understandable even to the non-clinical researcher, such as myself. This leads us into our question and answer, which is going to be moderated by Dr. Vince Faustino. So, Dr. Vince, yeah, hi.
Thanks, Michelle. And thanks, Jen and Phil, for those two wonderful talks. I think in the interest of time, because we want to be mindful of people's time, do we have questions from the audience? Let's start with that one so that we can get that out of the way If you have questions after this talk, I'm pretty sure Jen and Phil would be more than happy to answer them. But if you have questions right now, feel free to ask. I'm not seeing anything in the chat. Okay. Okay, so if something comes up, just give me a shout out, and then we'll go to your question. So the question now is, well, Jen sort of answered this question, but so I'm gonna revise this question a little bit. When would an RCT be preferred over causal inference study, for example, and the other way around? That's probably the better way of asking this, because this is not a dichotomy. When would you prefer one over the other? Let's start with Phil.
I guess I'd say if it was feasible to get the randomized controlled trial done in an efficient and cost-effective manner too, which is always um, the rate limiting factor. I guess it relates to feasibility, honestly. And if it can be done, it probably should, but it needs to be done right. And just because an RCT was done does not mean it was done well. If anything, the vast majority of the trials that are done, I think they compromise internal validity to help with recruitment rates, and then the trial is significantly flawed. Um, so if you're going to do it right. And if you can't do it right, do it Jennifer's way.
Okay, Jen.
I have exactly the same answer. Honestly, it's if an RCT can be done, if it can be done well, that's going to be preferred. The challenge in our field is that there are lots of questions for which I'm not sure that the RCT can be done or can be done well, given numbers of patients, and particularly when we get to the level of if we do start to understand those underlying biologic phenotypes and subdivide our already small patient populations even further. I think that the Bayesian adaptive trial designs helps this a little bit in terms of feasibility. And this is where I do see a lot of complement to utilizing the observational studies to more efficiently plan those trials and understand what those stratifications would be. And then you can take even better advantage of the Bayesian statistics that allow you to share information across groups and plan more efficient trials. But at the end of the day, there still may be some questions for whom you just don't have enough patients to put together a well-powered clinical trial. And that's where the observational causal inference is at least probably better than well, I did this last week and it worked. Let's try it again approach.
So, Jennifer, that reminds me of something that was incredibly unpopular when I said it at a policing meeting 3, 4 years ago, but that won't stop me from repeating it again, is that when we design trials, we should really consider enrolling all age groups and stop thinking about pediatric only trials up to 18 years of age, which has zero biologic relevance. And if we could be more successful at designing trials that included all age ranges, as long as it was appropriate for the question being asked, and then analyze age as an effect modifier to see, is there an age at which this therapy is helpful or not helpful or harmful? We are really shooting ourselves in the foot by only trying to recruit children less than 18 years of age for the recruitment reasons that you've mentioned. So I think it's something that we as a group need to be more open to and be willing to study adults at the same time to then allow us to ask the question, does age matter or not? And if it did, it might be 6 months of age. It might be 2 years. It could be never. And then you got to get faster.
Yeah, I just have like one pragmatic point regarding this, RCT versus non-RCT. There are interventions that are not used in practice. And therefore, there's just no way you can use observational data because the observational data doesn't exist. And sort of by default, you have to do an RCT. So those are the phase one, very early, new molecules. There's just no way doing causal inference studies for that. Uh, may, may, oh, Anil, are you gonna ask something? I see you
Uh,
unmute yourself.
if you have time, I was going to add to the discussion
Yes.
of causal inference by saying that causal inference is also, like the concept of heterogeneity applies there too.
Mhm.
All causal information inference is not same.
Yep.
Yes.
So rather than have a blanket statement of binary statement of yes or no, I would say it's a case by case evaluation based on both the benefit and harm kind of thing, like how difficult is it to do that intervention, how feasible or ethical is it to test that intervention, versus how robust is your causal inference methodology and results. So it is the same thing, one size doesn't fit all, the heterogeneity concept that Jennifer was talking about. So I think we can't give you a yes or no answer, but it is case by case, evaluate the methodology, evaluate the results, and then figure out whether it is necessary to take the next step or is it even possible.
Thanks, Anil.
Thanks. Couldn't agree more.
So for the last, just because we have like less, like about a minute to go, for our junior people, I just want to ask Phil and Jen if they are thinking about a career as a clinical trialist or causal inference person, how can they start? 30 seconds, each of you. Go, Phil.
Get a formal education in biostats, epidemiology, clinical trial design. Don't try to wing this. Get a master's or something similar to it to learn how to do it well.
Yep.
Perfect.
I'd
Jen?
say what Phil said, plus find a great mentor or a handful of great mentors, be a site PI. You learn so much about how trials work by being a part of a trial. And putting a plug out for Polisi as a really wonderful place to come and learn and collaborate and find mentorship. That'd be my four.
And
I
join
cannot
BloodNet
agree more. Yeah. Oh, BloodNet too.
if you're interested in that.
Yeah, I cannot agree more with what Phil and Jen said. Michelle, you want to have the last words? We're at top of the hour.
Yep. Thank you all for joining today. If you'd like to access this webinar as well as other webinars we've done before in the past on a variety of BloodNet topics, please join here or scan the QR code and I'll find a way to send this out to all the people who registered because a lot more people registered than were able to come this morning. So thank you to our speakers. Happy holidays, everyone, and happy Friday. Thank you.
Thanks, everyone. Bye, guys.